Hello students, welcome to Aakash. I'm your botany faculty Dr. Sukirti, and today we are here to discuss the solutions for AIATS Plus 2025 Phase 2 Mock Test 6, which was held on 23rd of Feb 2025, right? So in this particular session, we are going to deal with certain questions from certain chapters, as you have already done certain discussions in session 1 as well. So we are going to continue with the botany discussion in this session as well, right? And the topics that we are going to discuss would be the molecular basis of inheritance, anatomy of flowering plants, biological classification, right? So among these, if we talk about the molecular basis of inheritance, is a really important chapter because it actually gives us too many questions in many previous year papers as well, right? So let's quickly move ahead to the very first question we have, and that is, okay, so there is certain technical glitch, I suppose, so it's not working, but let's move ahead. Right, and the very first question over here talks about the heterochromatin differs from euchromatin as the former is, right? So when we talk about this heterochromatin and euchromatin, we should know what's the primary difference between them, right? So initially, agar hum ek cell ko dekhte hain, so in the nucleus, what we see is the chromatin fibers, the DNA present in the diffused form, right? Now, during the cell division, when the M phase has to be start, in that particular moment of time, this entangled mass needs to be condensed. And in order to do so, we require certain proteins, right? Till this level also, it's not like that the DNA is present without condensation, right? Because if we see the size of the DNA, it will be in meters, and the nucleus we are going to see would be in nanometers, right? So that is the reason why, to accommodate this much amount of DNA into very small nuclei, we need condensation. And in order to perform that, firstly, DNA, which is a negatively charged molecule, is going to wrap around a positively charged histone proteins, right? And when this is going to form, it is going to form nucleosome, right? And that's what you see in the interface nucleus, right? Over here, the bead on string structure, right? You'll be finding certain beads over here. So these beads are basically representing the nucleosome. So this condensation further requires certain, like after this linker histone, and further after this we need certain non-histone proteins as well, right, in order to perform this condensation. Now, the difference between these two lies in this packaging only. So when you will be having the condensation to a minimal extent, right, lesser condensation, then it will be euchromatin. When it will be more, it will be heterochromatin, right? So this is how we differentiate between the two. Now, if let's suppose we are saying that lesser condensation is there, that simply means that there will be loose regions of DNA that's also going to be available. That is the reason why when you stain this particular chromatin, it will be stained lightly, right? So it will be lightly colored one. So if there is a lesser condensation, we are going to call it as euchromatin, right? And because of this looser condensation, it will stain light too. So those areas are going to take up the stains. Similarly, because the DNA region is still loose, or we could decondense it back, right? So because of this presence of the looser region, we can also, accommodate certain proteins in order to perform transcription, right? So these enzymes could also access to these particular sites, and that is the reason why euchromatin is transcriptionally active, right? On the contrary, if we see heterochromatin, because it is really, the condensation is really high, right? Because of that, we don't have any free regions that are available for the accessing of the enzymes that are required for transcription. So this is the reason why it is transcriptionally inactive region. Right? And in the similar way, because the regions are not available, the stain is also going to be darker because it's going to, take up the surface part. So it's going to be stained dark as well. So it's a darkly stained region as well. Right? So these are the minimal differences between the heterochromatin and euchromatin, right? Now getting back to the question, if we see, the first one is saying that heterochromatin differ from the euchromatin as the former. So we should know about the characteristic of the former ones, right? So if we talk about heterochromatin, obviously it's more densely packed region of chromatin. So in a chromosome, if we have to localize them, let's suppose this is a chromosome we are talking about, we could localize this heterochromatin region over here in the centromere or maybe in the telomere, which is densely packed regions of chromatin. So this is true for heterochromatin, right? Then it says it is transcriptionally active, but that is incorrect with reference to heterochromatin. Heterochromatin, because of this dense packing, is not capable to provide the sites for the accessing of the enzymes that are required for transcription, right? So this is incorrect with reference to heterochromatin, but it is correct with reference to euchromatin. Now coming to without histone proteins, that is again incorrect. Histone proteins are the basic, or you could say the very first step of condensation of the chromatin material into, the DNA into the chromatin material and further condensation into the chromosome. So histone proteins are present in both of them, right? Then lightly stained region of chromatin. Again, as I have told you, euchromatin is the lightly stained region. Heterochromatin is the darkly stained region of chromatin, right? So this is how we could differentiate between heterochromatin and euchromatin. This question could turn out to ask you about the features of euchromatin also, right? So this is an important question which you have to study with respect to the differentiating analysis between these two types of chromatin, right? I hope this is clear to everyone. All right, now let's move ahead to the next one, which is question number 117, which says that what type of viruses are produced after the death of E. coli if a T2 phage having radioactive sulfur infects bacteria growing on normal nutrient medium, right? Okay, so this particular experiment talks about the experiment conducted by Hershey and Chase, right? So what are their objective? For any experiment you need an objective. Their objective was to identify that whether the DNA or the protein, which one of them is the genetic material, right? So in order to do that, they'll be requiring those two contender in a single organism. So that's why they have took a bacteriophage, right? So this particular virus has only two components. One is DNA and another one is protein, right? And another organism which can have its infection, so they have took another organism as bacteria, and that bacteria is E. coli, right? Now, how they are going to analyze this particular thing, that whether the DNA is the genetic material or protein is the genetic material. In order to do so, they have chosen the elements that are specific to these two macromolecules, right? Biomacromolecules. So we know for a fact that in DNA, phosphorus is unique, right? Phosphorus is found only in DNA. It's not being found in the protein. On the contrary, protein has sulfur, right? Now, where this phosphorus is present, it is present in the phosphate group of nucleotides, right? That's how the phosphodiester linkages are being formed. So DNA is flooded with these phosphorus molecules. Now, on the contrary, when we talk about protein, it has sulfur containing amino acids, like we could talk about methionine, we could talk about cysteine as well, right? And we have studied this, that methionine is the very first amino acid that is being recruited in the polypeptide synthesis, right? So that's why they have taken two different and unique elements that are being unique to DNA and protein, right? If they have performed this similar sort of experiment using the atoms like carbon or nitrogen or hydrogen or oxygen, right? If they would have taken, rather than these two, any of them, then they won't be able to differentiate that whether the inheritance or the transmission is occurring because of DNA or because of protein, because these four elements are going to be common for both of them, DNA and protein, right? So that's why purposefully they have took phosphorus and sulfur. Now they have made them radioactive, right? Because the usual elements, it's really difficult to identify them. That is the reason why they have took the radioactive forms of it. So they have took P32 to label the DNA molecules, and over here they have took S35 to label the protein molecules, right? Now, how they can label it? They'll allow the growth of these particular viruses in a medium that contains radioactive phosphorus, and on another plate they're going to have the growth in S35 containing medium, right? Now after this, what they have done? Now after this particular, labeling, they allow the very first thing to be done, and that's infection. Right? So when, let's suppose we are taking this and two sets, let's say this is the set in which DNA is being labeled and this is the set in which protein is being labeled, right? So let's suppose these are the E. coli cells, that's their original DNA that's being found, right? And as the E. coli cells are not being labeled, so they are going to remain like this only, right? Now, if let's suppose we are taking the viral particles, right? So over here, when we are taking the viral particle, okay, so let me just correct it up. So we are going to take the red things to say that these things are labeled. So over here, let's say this is the viral particle and its DNA is being labeled, right? In the second one, we have got the protein capsid of the virus that's being labeled, but the DNA was normal, right? So when they have infected it, at that time they didn't know that they only transfer the genetic material, right? So what they have done, they allowed them to infect the E. coli cells. So in this particular case, what happens after the infection is that this, after drilling it up, put the DNA here. Similar way over there as well, they have, inserted the DNA of these particular viral particles. But these viral particles are still being present, right, attached to it, attached to the bacterial cell. Now, in order to separate them, what they have performed blending, right? A mechanical thrush, because of which these two particular entities could be separated from each other. So, as a product of this, they have found out this particular thing and this bacterial DNA along with the labeled DNA, and in this particular case they have found a infected capsid, or we could say the labeled capsid along with this particular cell, right? Now how they are going to analyze that whether which particular material has been transferred? For that, in order to identify that, they have performed centrifugation. And we know in centrifugation, whichever is going to be the dense, or heavier particle, that's going to settle down, and the lighter ones are going to remain in the supernatant. And that is the reason why the bacteria, being bigger in size and heavy, is going to settle down in the pellet, right, over here. And over here in this particular section in the supernatant, we are going to get the viral particles, right. So, in both the cases, the same thing happened. This is the pellet that they have observed, and over here they will be having the viral particles. Now, what they have observed is that in this particular case where DNA is being labeled, the radioactivity is being found in the supernatant. But in case where the protein is being labeled, the radioactivity is being found in the supernatant, right. So, we know that what has traveled to bacteria in the pellet? It's the DNA molecule, right. So, this is how they have distinguished and provided an unequivocal proof that DNA is the genetic material, right. Now, getting back to the question, this question is saying that what type of viruses are going to be produced after the death of E. coli if a T2 phage having radioactive sulfur infects bacteria growing on normal nutrient medium, okay. So, when the protein is being labeled, so the question is explicitly telling you that the T2 phage is labeled with sulfur, that means the capsid is being labeled, but the DNA present inside it is entirely normal, right. So, we know after the infection, this viral capsid is going to be a separate thing, right, a separate molecule, and the entire new viral particles are going to be made out with this particular DNA, right, the normal DNA. Because virus is a parasite, it's going to use the machinery of the host cell, and in order to direct the synthesis of its viral capsid, it is going to utilize this DNA, which is entirely normal. That is the reason why if this particular bacteria, right, later on allows to be grown on the medium and the T2 phage particles are going to burst it out and come out, then all those viral particles will be having normal capsid and normal DNA, right, because this particular labeled one has already been separated from it, right. So, and if this particular question is asking about that what is going to happen if it is being labeled with the radioactive phosphorus, in that particular case what we are going to see is the radioactive viral particles, right. The T2 phage, even if they are growing on the normal medium, then also they will be having certain amount of radioactivity. Over here they won't be having any radioactivity if they are being transferred to the normal medium, right. So, this is how we have to interpret it. So, getting back to this particular one, this is saying that what type of viruses are produced after the death of E. coli if a T2 phage having radioactive sulfur infects bacteria growing on normal nutrient medium. So, the first one is saying it will be radioactive capsid and non-radioactive DNA. As I have told you, because the radioactive capsid has already been separated out from the bacteria, so that is why after the growth on a normal medium, it does not have any radioactive source, right, and that is the reason neither the capsid nor the DNA is going to be radioactive. Then the second one is saying no S35 in protein coat but radioactive sulfur in DNA. This is also incorrect. Why? Because when we talk about sulfur, it is not being present in DNA, right. Then non-radioactive capsid as well as genome, so that is correct, right, because the virus is going to use DNA as the dictating molecules to assign that what kind of assembly should be there with the viral capsid. So, that is the reason why both capsid and genome is going to remain non-radioactive. Then the fourth one is saying radioactive phosphorus and sulfur in DNA as well as in capsid. That is again incorrect. We are not even providing the radioactive phosphorus, how it is going to be present in the upcoming viral particles, right. So, this is how when we analyze it, the correct answer for this particular question is going to be option number 3, right. I hope this is clear to everyone. In case you have any doubts, please put up in the chat box, okay. Now, let us move ahead to the next one, the next question, and it says that which of the following correctly represents central dogma of molecular biology. So, when we talk about central dogma of molecular biology, it talks about that how exactly the genetic material is going to express itself, right? And this particular proposition, that this central dogma proposition, is given by Francis Crick, right? Of course, he's been associated with proposition of the double helical model of DNA as well, along with James Watson. He has certain other contribution as well, like he also postulated the presence of an adapter molecule, which can be used during translation. He also gave the idea of the semi-conservative nature of DNA replication as well, of course, along with Watson, when they are proposing their double helical model of DNA. So Francis Crick has proposed this particular proposition of central dogma of molecular biology, and in this particular dogma starts with the genetic material which is present in almost all cellular organisms, that is DNA. So it always starts with DNA. So as option number 3 is starting with a DNA, that is the reason why we are going to choose this particular option, and we could rule out all others, right? Then DNA is going to convert itself into mRNA, right, and then further into protein. So ideally, initially what DNA does, it makes its copies by the process of replication, right, making its copies. Then further it will perform transcription to make this mRNA, and finally it performs translation to make proteins, right? It is similar like that if something is being written like rigorously, right? Like for example, if some scientist is writing his research papers, so that research paper is going to have certain holding or application, right, in the near future. But then of course it will be really difficult for a student like you, like in school, to interpret it, right? So DNA is that complex thing, and protein is the one, you students who have to like literally make it happen. So the teacher in between, this mRNA is basically solvifying or making it easier for you to understand. So similar way over here, mRNA formation is basically decoding this DNA information into a simpler form so that it could translate itself into proteins, right? So this is the central dogma which is proposed by Francis and Crick, and over here it's correctly being depicted in option number 3. Right? All others could be ruled out because they are not even starting with the genetic material, right? In the previous question itself, we have solved this particular dilemma that whether protein is going to be the genetic material or DNA is. So protein is not the genetic material. So and this particular one is starting with it. That is the reason why it's incorrect. Over here, they are starting it with the mRNA, which is a product of transcription from the DNA, right? So mRNA cannot be produced if DNA is not being there as a template. So that is why we can rule out all others, and the correct answer for this particular question is going to be option number 3, right? So yes. All right, moving ahead, we have another question which talks about that the distance between two consecutive base pairs according to Watson and Crick model of DNA is, right? So if we talk about the model proposed by Watson and Crick, that is the double helical model. Now, this particular model was so accurate according to the techniques that has been present at that time, that it has ascribed all the characteristic of DNA to us. So according to this particular model that was proposed by Watson and Crick, firstly, the double strand, it is for the double stranded DNA, they have said that the backbone is going to be made up of sugar and phosphate, right? And the nitrogenous bases are going to be present inwards to it, right? Another thing that they have proposed is that the two strands are going to be anti-parallel to each other, right? They are going to be anti-parallel. That means if one strand has got the polarity of 3' to 5', another one is going to have the polarity of 5' to 3', right? Another proposition of this one is the base pairing, which is a hallmark for this particular double helical model, that they have proposed that the adenine is going to pair up with thymine and the cytosine is going to pair up with guanine, and these pairings will be done with the help of hydrogen bonding, right? They also said that the stacks, the base pairs, right, different base pairs stack over one another. Right? So these particular propositions, like the presence of this hydrogen bond, this stacking, like if this is the one base pair, another one is going to stack over it. This stacking is what actually gives the double-stranded DNA its stability, right? Concept of lac-operon. Okay, so after this we'll get back to that particular one. All right, so so this particular stacking and the hydrogen bonding is the thing which is basically providing the stability to the double helical model of DNA, right? So these are certain propositions. Along with that, they have also defined the pitch of the DNA. That's going to be 34 angstrom, or in nanometers it will be 3.4 nanometer. They also said that in one turn we'll have 10 base pairs, and the distance between the base pairs would be 3.4 angstrom or 0.34 nanometers, right? So these are the propositions, and because of this accurate understanding of the structure of DNA, they're also awarded with the Nobel Prize, right? So this model is really a hallmark in the scientific field, that it has accurately describes that how exactly the DNA structure would be aligning itself. So now with this particular information, if we get back and identify, they are asking that what will be the distance between two consecutive base pairs, right? So according to this particular model, it was 3.4 angstrom or 0.34 nanometer, which is correctly depicted in option 2. This is incorrect, 3.4 nanometer. Usually what we do is we often get confused between angstrom and nanometers, right? So it will be it will be different in terms of angstrom and nanometer. Nanometer is usually 10 to the power minus 9 scale, while angstrom is 10 to the power minus 10, right? So over here, this particular option, option number 2, correctly depicts the distance between the two consecutive base pairs, right? So this is how we'll be solving this particular question. So now as the student is asking about the lac-operon, okay, so let's quickly revise that particular concept. So it is hallmark of double helix model is base pairing only. No, the presence of hydrogen bond as well, and the complementarity that they have defined, that is also the hallmark. Rest all the anti-parallel, see, hallmark is certain thing which is so unique that nobody else was ever thought of it, right? So that is the reason why we say that base pairing and hydrogen bonding that they have proposed were the hallmarks of their model. Also the structural analysis, but structural analysis could be later confirmed, or you could say it is also a slight bit of rough data was also provided by Rosalind Franklin and Morris Wilkins as well. Not the accurate data, but the slightly rough data. So the hallmarks that entirely being done by them was the base pairing and hydrogen bonding, right? That's why we constantly pronounce it like that, right? Is it clear, Shubhro? Okay, so till that particular time, let's quickly revise the lac-operon thing as well. So when we talk about lac-operon, so obviously there will be like a sequence of genes. They are going to function in conjunction with each other so that they can control the metabolism of one particular molecule. Over here, that molecule is going to be lactose, right? So if we can see the entire operon, it's gonna be like So these are, let's say, the structural genes. Obviously, the terminator is going to be there. So Y, Z, it's the operator region, this is the promoter region, right? Now, what is an operator region? Operator region is something which is going to overlap with the promoter, but this is the site for the regulatory molecules to bind, right? Now, upstream to this, we have the repressor gene along with, of course, its promoter, right? And terminator is going to be here, because in a transcriptional unit you have read this already, that you will be having a promoter, set of structural gene, and then you will be having the terminator, right? So firstly, when the lactose metabolism has to happen, when it is present inside the cell. If it is not present inside the cell, metabolism is not required, right? Its lysis, its catabolism is not at all required. We know that lactose is a carbohydrate source which could provide us with energy, or can provide us with the monosaccharide molecules that can further help in performing the glycolysis or the pathways of aerobic respiration, right? So firstly, let's consider that if lactose is not present inside the cell, in that particular case, what happens is, because this is firstly present upstream, right? Also because of the smaller size, it's gonna transcribe first. So this is going to create its mRNA. After the transcription is completed, the translation will occur and a repressor molecule would be formed, right? Now, what this repressor molecule will do? It will go and bind to the operator region like this. Now, in order to perform the transcription of ZYA, similar like this repressor gene, what we need? We need the binding of the RNA polymerase to the promoter region. Over here, because this promoter region is free, RNA polymerase can easily bind to it and can perform its transcription. But here, as I've told you, this operator region is basically overlapping with the promoter one. Now the RNA polymerase does not have the site, like the entire site to bind. That is why this particular binding is not at all possible. And now, because this binding is not going to take place, right, then do you think that the transcription is going to be taking place over here for these particular structural genes? Of course not, right? So, agar transcription is not going to take place, of course translation is also not going to be there. And that is why these three structural genes won't be able to express itself, right? But this particular operon is a bit, leaky in the sense that, let's suppose 100 times this particular thing has to express itself. 99 times this particular expression is not going to take place. Maybe one time what happens is that prior to the binding of repressor to the operator, RNA polymerase already binds to it and transcribe it. So this minimal expression is required, right? This minimal expression is required. Why it is required? Now we will understand. In the case when the lactose is going to be present in the medium in which the bacteria is growing, right? Now we are considering that the lactose is being present in the system, right? So if lactose is being present in the system, in that particular case, again this particular gene is going to express itself, going to form the repressor protein, right? But now because lactose is present inside the medium and therefore can enter into the cell, now this lactose is going to bind to the repressor and going to cause a conformational change in the repressor in a way that it won't be able to bind now to the operator, right? Even if it is reaches in the close proximity, now it does not have that particular, shape, specificity for this operator to get binded with. So that is the reason why this promoter is going to remain free for the binding of RNA polymerase. Now when RNA polymerase binds to this particular promoter site of these structural genes, it will allow its transcription to occur, right? As it is forming a single mRNA molecule for this entire stretch, that is how we know that it's a polycistronic gene, multiple cistrons in one particular stretch, right? So they'll be having multiple, they are going to code for multiple polypeptides, but the mRNA that's going to be formed will be one, right? After this, as the mRNA is already being formed, now the translation will start. And from this Z section, we are going to have the formation of beta galactosidase, right? With this Y, we are going to have the formation of permease. With this A, we are going to have the formation of trans-acetylase, right? So this beta galactosidase is responsible for the breakdown of lactose, which is a disaccharide, into its monomeric units, which is glucose and galactose. This permease is required to allow the entry of lactose and the beta galactosides in the cell, right? So that is the reason why I have told you that this particular operon is a bit leaky, where it will allow the minimal expression of these particular structural genes even in the absence of lactose, right? So this is not being mentioned in NCERT, that's why we are going to consider this particular case always, that if the lactose is not present in the medium, there will be no expression. If there will be lactose in the medium, there will be expression, right? So why I am telling you that particular detail is because when permease is not there, how the lactose is going to enter into the cell. So in case initially a little bit of lactose which is present in the medium could enter into the cell, this permease is required inside the cell, and because of that, that minimal expression is present, right? But because we are preparing for NEET and we have to go by the standards of NCERT, so what we are going to study that if lactose is not present, then there will be no expression of structural genes. If it is present, there will be the expression, right? So I hope this is clear to you, Shubhro. In case you have any doubt, you can put in the chat box. All right. So let's move ahead to another question, which is from biological classification. And it says that we have to match column 1 with column 2 and we have to choose the correct option. And over here in column 1, we can see the protozoans, right? So when we talk about the protozoans, the first common feature in all the protozoans is that they lack cell wall, and that is why they are believed to be the ancestors of the animals, right? Because they don't have it. Also, they are heterotrophic in nature, right? We don't have the autotrophic species present over here in this. And these characteristics are also being found in the animal kingdom as well, which are entirely heterotrophic. Also, they don't have the cell wall in their cell, right? So when we talk about protozoans, basically they are being classified on the basis of like what kind of locomotory structure that they possess, right, among the other features. But this is a prime basis on which we could classify these protozoans. So we can have the amoeboid protozoans, we can have flagellated protozoans, we can have ciliated protozoans, and we can have sporozoans, right? So now when we talk about the locomotory structure that we can find within them, over here in amoeboid, like it simply means they are amoeba-like, right? We have studied amoeba a lot many times, that it will be producing its false feet, that's pseudopodia. So generally amoeboid protozoans have the locomotory structure as pseudopodia. So they move and they also capture their prey by this particular structure known as pseudopodia, right? When we talk about flagellated protozoan, as the name itself suggests, the locomotory structure that they're going to possess would be flagella, right? In ciliated, it would be cilia. Now when we talk about the sporozoans, these organisms are the ones that do not have any locomotory structure, right? That's absent in them. Now when we talk about the amoeboid protozoans, right, so they'll be having this specialized feature as pseudopodia. Also what we found is that in some of their marine forms we can find the coating of silica on them as well, not in the cell wall, right? So don't confuse it with diatoms, which also have silica, but those organisms, the diatoms, have the silica in the cell wall. Over here they don't have any cell wall because they belong to the group protozoan, right? So over here they can found the silica as the coating on their outer surface, right? Then when we talk about the flagellated protozoan, what's the most important thing on which the question comes? The organism that belongs to it, that is trypanosoma. This particular organism is responsible, or we could say is the causative agent of sleeping sickness disorder, right? So this is a question that's been frequently asked, right? So this is one of the examples of the flagellated protozoans. In amoeboid protozoans, of course, the example would be amoeba or antamoeba, right? Then when we talk about the ciliated protozoans, cilia, along with having these moving structures, cilia, right, which are minute than flagella, they also have a body cavity known as gullet, right? So that is a special feature with the ciliated protozoans. And when we talk about the organism that can be categorized in this particular group, then we could take the example of paramecium. Right? Then talking about sporozoan, which do not have any locomotory structure present in them, but the organisms that are being present over here, they have this infectious life cycle, which is going to be requiring, you know, certain host. Like we could see the example of malarial parasite, which is Plasmodium, right? It belongs to this particular category. So now based on this, if we go back and see the question over here, it says that flagellated protozoan, right? So what does flagellated protozoan have? As I have told you, the organism that belongs to this particular class is trypanosoma, which cause the disease sleeping sickness, right? So over here we could clearly match A with 3, right? And as A could be matched with 3, we could rule out option number 2 and 4, which is saying that A is going to be matched with 2. What 2 says that it has false feet to capture its prey. False feet means they are referring towards pseudopodia. Pseudopodia is not being found in flagellated protozoan. On the contrary, they are being found in amoeboid protozoan, right? Now, the B1 is saying ciliated protozoan. So, as I have told you that they have a specialized cavity within their body that's known as gullet. So, they have the cavity that opens to outside of the cell surface. So, they are referring towards the gullet. So, B matches with 1 over here. Right? Based on that, we could rule out option number 1, which is matching B with 4. Right? 4 is saying that it includes organisms that have infectious spore-like stage. Right? Generally, they don't have the things like this. Now, amoeboid protozoan. So, when we talk about the amoeboid protozoan, so they have false feet to capture their prey. So, C matches with 2 and D says it includes sporozoans, which includes organisms that have infectious spore-like stage. So, D matches with 4 and the correct depiction you could see in option number 3, right. So, this is how you will be solving this particular question. Also, the type of protozoans are important because many easier questions could become from this particular topic of the protist, right. I hope this is clear to everybody. Now, moving ahead, we have another question which says mycoplasma has. Right. So, when we talk about this mycoplasma, so firstly, this is a prokaryote, right. This is really important to understand that it belongs to the kingdom Monera, right. So, Monera is a kingdom that has been taking up or grouping up all the organisms that are prokaryotes, right. So, this particular kingdom is being specified for these organisms. So, mycoplasma is being included over here, by which we could clearly infer the fact that mycoplasma are basically prokaryotes. So, what is being different in mycoplasma which is being present in bacteria, like why we are not having the same name for it? It is because of the fact that being a prokaryote, that's correct, but unlike the bacteria, if we say that they don't have cell wall. Bacteria does have it, but the mycoplasmas are not having it, right. So, what they lack is that they lack cell wall. This is one of the primary features, right. Cell wall is absent in them. Another important, role if we talk about the mycoplasma is that mycoplasmas can live without oxygen. Right? They can live without oxygen, and also they are pathogenic to animals as well as plants. Right? So now based on this, if we talk about that what all characteristic mycoplasma is going to have, so it said mycoplasma has. So firstly, it has nucleus? No. Why? Because when we say that mycoplasma is a prokaryote, we know for a fact that prokaryotes are the organisms which do not have a well-defined nucleus. On the contrary, they have an irregular, or we could say a region where the genetic material is being concentrated, and that region is known as nucleoid, a nucleus-like region, not nucleus, right? So we could rule out this particular option. Then option 2 says cell wall, as I've told you explicitly that how they are differing from bacteria, because they do not have the cell wall, unlike bacteria. Now option 3 says 70S ribosome. So this is correct. When we talk about mycoplasma being prokaryote, so prokaryotes do not have the compartmentalization in their cell. So that is the reason why whatever ribosomes they are going to have, they are going to retain in the cytoplasm. So their cytoplasmic ribosomes are of size 70S, right? In which the smaller subunit is of 30S size, while the larger one is 50S size, right? So ribosome is the non-membrane bound organelle, and this is an organelle that is common between the eukaryotic and prokaryotic cell. 70S ribosomes are also being found in the eukaryotic cell, but there they are not the cytoplasmic ones. On the contrary, these 70S ribosomes are being present in the plastids or in mitochondria, right? Now option 4 says single-stranded DNA. That's also incorrect. So this being a cellular organism, so cellular organisms have double-stranded DNA as their genetic material. Mycoplasma is not the exception. So that is why we could rule out this option as well. So mycoplasma among these particular structure is going to have 70S ribosome. So for this particular question, the correct answer is going to be option number 3, right? I hope this is clear to everyone. All right. So now let's move to the next question, which is question number 98, which says that at maturity, which component of phloem is dead, right? So when we talk about phloem, this is a food conducting tissue, right? And that is the reason majority of its components are living in nature. So phloem has 4 components. The first one is the sieve tubes. Another one we have companion cells. Then we have phloem parenchyma. And then we have phloem fibers. Right? Now when we talk about the sieve tubes, these are the cells, like it's an elongated structure, and the cells that are constituting it would be the ones that are enucleated. They don't have nucleus in them as such, right? And they have peripheral cytoplasm and a very large vacuole. Then in the companion cells, we does have the nucleus, and what we know is that when we talk about the nucleus, it controls the activity of the cell. As these particular ones are lacking it, that is why their activity is going to be controlled by the cell which is carrying it, and those particular cells are companion cells. So companion cells regulate their activity, specifically they maintain the pressure gradient in the sieve tubes. So these two cells together is regulating the food conduction in the plant, right? So that is why together they are also being known as the sister cells, but these two are living cells, right? Then we talk about phloem parenchyma, again a living cell, right? Parenchyma is composed of the living cells only, and this phloem parenchyma is usually not being found in most of the monocots. And then the last structure, which is phloem fibers, right? So these are the only ones that are the dead components, right, at maturity. So these dead fibers, why? Because they are sclerenchymatous in nature, that is why we can consider them as dead, and these are the only component of phloem that are dead. All else are the living components of phloem. So as the question is asking that at maturity, which of the component is going to be dead? That is going to be phloem fibers, given in option 2. Option 1, phloem parenchyma, companion cells and sieve tubes are going to be the living one, as they have to conduct the food in the entire plant body, right? So this is how we could be solving. It's an easier question, but certain times they can ask you the question regarding their structural details or maybe the function that they perform. A very common one is regarding the phloem parenchyma. Multiple times it's been asked that what is being present in the monocot stem or dicot stem, right? Or what could be the differentiating feature? This would be the differentiating feature, because in dicot stem you're going to see it, while in the monocots you're not going to see it, right? Moving ahead, we have another one that talks about that, read below the given statements of assertion and reason, and we have to select the correct option. So assertion over here says that water molecules are unable to move across the endodermis of root in the areas of Casparian strips. So this is the correct statement, because when we talk about the water transport, so let's suppose this is an epidermal cell, right? So let's say we have got an epidermal cell, then we have a cortical cell, then we have an endodermal cell, right? Further the pith, and finally xylem, right? Let's suppose we have got these sort of things. So these are the cell wall regions, and then we have the internal living components present here. So let's say this is the epidermal system, this is the cortex, the endodermis, and this is the pith region, and finally it is going to xylem, right? So when we talk about this water transport, and if the water is like moving like this, right? So over here in endodermis, we have got this Casparian strip, and these are the regions that are the barriers for the water transport, as this particular layers are completely impermeable for the water molecules, right? So that is why these are being blocked in these particular regions in the endodermal cells. And that's what the, statement in the assertion is also saying, that they are unable to move across the endodermis of root in the areas of Casparian strips. So that is correct, because they can move via this particular living pathway easily, but if they are moving towards this, if they are taking this apoplastic pathway, then they are going to be blocked by these Casparian strips. Now why they are being blocked? Because the Casparian strip is made up of a material that is known as suberin, which is a waxy material, and it is impermeable for the water molecules. That is the reason why this particular passage is being blocked, right? Now moving to the reason statement, it says that Casparian strips are special thickenings of water impermeable waxy material suberin in the radial and tangential walls of endodermal cells. So this is also correct, right? They are being found in the radial and tangential walls of endodermal cells, and they are being, made up of this water impermeable substance known as suberin, right? So that is what I've explained you as well, that why the water transport is being blocked, why the water is unable to move across it, because it is made up of a material that doesn't allow the movement of water. So the assertion is also true, reason is also true, and reason is basically explaining that why assertion is going to hold true. That is the reason why we are going to choose option number 1 as the correct answer, and we could rule out all others. Right? So the correct answer for this particular question is going to be option number 1, right? So this is how you will be solving the questions of anatomy and the molecular biology, right? And I hope this session is useful to you. So stay tuned with this particular session. Thank you very much for your time.